So today we're so pleased to have Federica Vecchio, PhD, joining us to present investigating the impact of Coxsackievirus infection on beta cells and the corresponding antiviral CD8 positive T-cell responses. She's coming to us from INSERM Cochin Institute in Paris, France. And she studied medical biotechnology, a little bio, studied medical biotechnology at University of Milano, Brescia, Italy, where she received an MSc in 2018. In 2016, already before MSc graduation, she got involved in T1D research at the DRI of San Raffaele in Milan, in Italy, where she started working on T1D pathogenesis, exploring the role of neutrophils in disease development. And in 2019, she moved to Paris, France, to start her PhD as a member of the Malone and Yu laboratory team at INSERM Cochin Institute. The interplay between Coxsackievirus infection and the immune response in T1D progression has been a focus of her project. And after receiving the PhD in 2023, this topic remains the core of her present research work, which she is further exploring as a postdoc. The excellent and exciting paper that just came out is titled Coxsackievirus, infection induces direct pancreatic B-cell killing, but poor antiviral CD8 positive T-cell responses. And we're going to drop that in the chat along with instructions about asking questions. Thank you so much for joining us today, Federica. I really appreciate you making the time and look forward to this interesting talk.
Thank you, Monica. Hello to everybody. Today I'm going to present you how we investigated the impact of Coxsackievirus infection on beta cells and the corresponding antiviral CD8 T-cell response. So as you know, the type 1 diabetes natural history is divided into disease stages, and what is commonly recognized as type 1D is actually the latest stage, which is the stage 3, in which we have the onset of clinical symptoms, which are described as the 4 Ts. And then in the long-standing disease, we have the possibility of the development of long-term complications. So this is why it is important to intervene as soon as possible with glucose monitoring and insulin injection, as it has been shown that improved glycemic control decreases the development of complications over time. So this is an overview of what is commonly recognized as type 1D, but today we're going to focus on how do we get there. Before this stage, we have the stage 2, in which dysglycemia appears, meaning that we have an impaired glucose tolerance that can be measured with an oral glucose tolerance test. And before this stage, we have the stage 1, in which the tolerance to glucose is still normal, but the autoimmune process is already ongoing, meaning that we have the presence of autoantibodies. And it has been shown that the higher is the number of autoantibodies, the higher is the probability to develop type 1 diabetes. So during these pre-symptomatic phases, as you know, we have immune infiltration in the pancreas that is characterized at the very beginning by main infiltration composed by innate immune cells, and at the latest stages composed mainly by B and cytotoxic CD8 T cells together with CD4 T cells. And here we have an example of what is known as insulitis, so the infiltration around the islet in the pancreas. So today we're going to focus at the very beginning, so the stage 0, which represents the real starting point of the disease. At this stage, we have genetically at-risk population and the presence of a trigger that makes the disease begin. We have the presence of genetic factors that provide the risk for type 1D development, and we can say that the HLA class 2 and class 1 region accounts for almost 50% of the genetic weight in type 1 diabetes, together with more than 60 known HLA variants that have now been linked to type 1D risk. However, genetic factors are insufficient to explain the majority of type 1D cases, and this is due to many reasons. Here we have some examples of observations that have been made. So first, the left lung type 1D concordance in monozygotic twins is up to 72%. Second, another example is given by immigrants. So we have epidemiological studies that showed that immigrants typically get the same type 1D risk as the native population of the place where they go. And the fact that the type 1D incidence is increasing while the prevalence of predisposing HLA alleles in new onset patients is decreasing. So if on the one hand we need to consider a substantial genetic determinant, on the other hand we need to keep in mind that there is a major increasing role played by environmental events. So among these environmental events, enterovirus infection, and in particular Coxsackie-virus infection, has been shown as one of the major candidate environmental triggers for type 1D development. So what are we speaking about? Coxsackie viruses. Coxsackie viruses are small enteroviruses that are composed by a single-strand positive sense RNA genome. And here we have the scheme of the genome and the representation of how the capsid is composed. So here we have the several proteins that are inside. And these viruses use the mucosal tissue of the digestive or respiratory tract as the main site of replication. But the way CVB binds to host receptor, so to enter the cell, one of the receptors is the Coxsackie adenovirus receptor, the CA receptor, which is expressed by beta cells, among others. So CVB infections are common infection, especially during childhood, and usually they lead to asymptomatic or cause cold-like symptoms, so very mild symptoms. However, the enteroviruses can progress from this stage to attack a particular organ, so the brain, the heart, the pancreas, so we can have the development of serious complications such as meningitis, encephalitis, myocarditis, or pancreatitis. And it is important to keep in mind that infections of the heart or central nervous system can be fatal. So, what about vaccination? Well, we need to mention the PRV-101 phase 1 safety trial based on CVB vaccination led by Prevention by Sanofi, and this phase 1 has ended and has demonstrated the vaccine to be safe and efficient in stimulating the production of neutralizing antibodies against the Coxsackie-virus, so both at high and low dose. So Coxsackie-virus infection is associated with type 1 diabetes. When we speak about this association, we have a temporal correlation, meaning that the prolonged exposure to the prolonged presence of enterovirus B serotype has been correlated with the seroconversion to autoantibodies. And also a special correlation, meaning that the presence of VP1 positive cells have been found in, this is the viral protein, has been found in the islet in the pancreas. And the presence of VP1 positive cells has been shown to be higher in terms of percentage of the total islet in type 1D organ donors versus autoantibody positive and non-diabetic organ donors. However, we only have little data about the effect of Coxsackie-virus and about the anti-Coxsackie-virus T-cell responses on beta cells. So we wondered what was at the basis of this possible causal relationship between Coxsackie-virus infection and the development of type 1 diabetes. So we imagined three possible but non-exclusive scenarios that I'm going to present you. The first one is a direct lytic effect of the virus on infected beta cells with a consequent release of self-antigen characterized by a poor Coxsackie-virus antigen presentation and a poor anti-CVB immune response. Or we might have an indirect lytic effect of antiviral immune response on infected beta cells, again with the self-antigen release, characterized by a strong CVB antigen presentation and a strong anti-CVB immune response. Or we might also have epitope mimicry mechanisms at play, which means that physiological antiviral immune response might shift into a pathological anti-islet autoimmune response. So we try to answer a few questions. First, how do beta cells behave upon Coxsackie-virus infection? Next, how is the T-cell response mounted against the Coxsackie-virus? And what is the relative contribution of Coxsackie-virus on one side and CVB-reactive T-cells on the other in beta cell killing? So here I'm going to show you the results that are included in the paper that we just published on Science Advances, and this is the very beginning of all our experiments. So we try to understand how do beta cells behave upon Coxsackie-virus infection. We took some beta cells line, so we used the ECN90, and we infected them with different strains of Coxsackie-virus. Then we followed them at various time points to understand which was the best one to have a good viability and a good amount of cells that were positive for VP1 and double-stranded RNA, meaning that they were infected. And we picked the 6 hours time points for the other experiments. So we measured the HLA expression that was on these cells that were exposed to the virus, and we could observe that we had a HLA class 1 downregulation upon infection. Also, we identified the HLA-bound peptides by mass spectrometry, and this was the part of the work done by Alexia Carré, previous PhD candidate in the lab that developed her project on this side. And we could identify that only few CVB peptides are presented on the surface of beta cells upon CVB infection. So we took these few CVB peptides that we could identify, and we put them together with some in silico predictions that were made in collaborations with Isaac Snowhite. And we ended up with some HLA-A2 and HLA-A3 restricted peptides to add to our eluted peptides. So at this point, we wanted to understand what was the T-cell response and how this T-cell response was mounted against these CVB peptides identified. So we used the recombinant HLA-A2 and HLA-A3 multimers that were loaded with several peptides, and we used a combinatorial analysis, meaning that, as you can see in this example, each population of CD8 T-cells recognizing a peptide was identified by a single combination of color of fluorochromes. So as you can see in the top right corner, you have the name of the peptides that were tested, and here you have the flu that was our control. And in the bottom right corner, you have two numbers. The first one is the frequency that was measured on the total amount of CD8 T-cells collected on each sample. And the second one is the percentage of cells that were positive for antigen experience marker. So here you see the results put together on the left two graphs. You see the HLA-A2 panel, and on the right ones you have the HLA-A3. And in the top ones you have the frequency of these cells recognizing CVB peptides. Each column is a different peptide, and the last one is the flu. And in the bottom two, you see the percentage of effector memory of these cells. So as you see, among all the peptides that were tested, we could observe that when comparing this response against the response that we obtain for our control, the flu peptide, we could observe that the overall CD8 response against Coxsackie-virus was quite poor. So we also observed that some of these CVB reactive CD8 T cells were actually cross-recognizing a homologous GAD epitope. Here you have the CVB peptide that was considered and the GAD epitope, and the regions of this peptide was already known in 1994, so we could understand which was the specific peptide identified. And we used the same method to analyze how many of these cells per sample were recognizing GAD or the CVB peptide alone, and how many of these cells were cross-recognizing both. And as you see in this graph, in the first column you have the CVB alone, in the second one the GAD alone. And as you can see, overall the number of CD8 cells cross-recognizing the two was higher than the other two. So suggesting that actually a possible cross-recognition is at play between this specific CVB peptide and the GAD epitope. However, the results that I showed you so far were obtained from experiments performed from adult blood samples. So we wondered whether this low response was due to this long distance since the time of the exposure to the virus, because as I mentioned before, Coxsackievirus infections are especially common during childhood. So we decided to explore what was the response also in blood samples from children. And the first problem that we had to face was the amount of blood that we were getting, because in any case, this is an analysis of a rare population, relatively rare population, and we needed a certain amount of blood. So the first point was to develop a method that was reassuring you that our data were reliable, meaning that we identified which was the coefficient of variation and how this coefficient of variation was increasing among the various observations made on a specific amount of CD8 T cells according to the amount of T cells that were tested. So we defined a minimum number of CD8 T cells, and the samples that were not satisfying this cutoff that we defined were not included in the analysis. So when, after applying this method that was developed in collaboration with John Cadiz, we could have a look at what was the result on blood samples from children. And we could see that, again, we had quite low response in terms of CD8 T cells recognizing CVB. And this was valid when the analysis was performed on healthy children and on type 1D children. So we could conclude that the response of CVB was the low response of CD8 cells against CVB detected in the adults was not due to the long time since the moment of the exposure to CVB. We wonder whether this low response was specific for the circulating CD8 T cells. So we wanted to understand what was the situation in lymphoid organs. So we studied the CVB-reactive CD8 T cells in human lymphoid organs that were provided by the Network for Pancreatic Organ Donors with Diabetes and POD, which provided some spleen materials on which we could analyze the presence of these cells. In these graphs, you see that we could work with the type 1D organ donors, autoantibody positives, organ donors, and non-diabetic. But the point was not to compare the various cohorts, mainly because of the numbers. Overall, what we could observe was that the frequency, that again is in the top two, and the percentage of effector memory in the bottom two graphs. The frequency was quite similar, but we could observe that the percentage of effector memory in those cases in which it was possible to define this percentage, meaning that the cells were enough, well, this percentage was almost always extremely high, and this was something different from the blood. And also we explored some more markers, and we analyzed the percentage of CD8 T cells that were positive for PD-1 and negative for CD25. So when comparing cells that were analyzed from the pancreatic lymph nodes, cells, CVB-reactive CD8 T cells against the flu-reactive CD8 T cells from the same donor, we could observe that in the first case, this percentage was higher. So this suggested that these cells presented an exhausted-like phenotype. And we proceeded with the sorting of these cells because we wanted to perform some TCR sequencing. In this case, you can see in this graph 3 donors from which we analyzed different tissues. So for the first donor, which is represented by the first 2 columns, we have the spleen and the pancreatic lymph nodes. From the second one, blood and spleen, and for the third one, blood and pancreatic lymph nodes again. So when comparing the results from the TCR sequencing, we could find some of these sequences that were actually repeated, but only within the tissues deriving from the same donor, suggesting the presence, the existence of private, not public clonotype, meaning not shared across various individuals. And in collaborations, thanks to our collaboration with Teresa Rodriguez Calvo, we also performed some stainings to identify these cells, these CD8, CVB reactive CD8 T cells in the tissues. And as you see, it was possible to observe, to study several tissues, such as spleen, pancreatic lymph nodes, and the pancreas. And when looking at the pancreas, when looking at CD8 T cells recognizing CVB, we could conclude that also in the tissue, in the pancreas, the number of these cells was quite low, suggesting that, as in the blood, the CD8 T cell responses against Coxsackievirus are limited also in the pancreas. So at this point, we wanted to understand what was the behavior of beta cells upon CVB infection, as we could observe a quite low response in terms of CD8. We wanted to understand better what was the amount of beta cells killing mediated directly by the virus. So we took some viable beta cells and we infected them with GFP CVB, and this made it possible to follow the infection because we could see cells becoming fluorescent when they were infected, and in this case GFP positive. And then with the use of a dead cytotoxic red marker, we could identify cells that were dying. So this is what we obtained after observing the cells for more than 48 hours and acquiring images with live imaging techniques, which is the IncuCyte, the instrument that we used. And we tested several amounts of virus. And here you see MOI 300 until MOI 10, the lowest in this experiment. And you see the various shades of red, of green, representing the peak of the infections, and the various shades of red representing the peak of dead cells. And we could observe that for all the conditions, around 20 hours later than the peak of the infections, almost all the cells were dying. We also made the important observations that Coxsackievirus, once inside the beta cells, is able to transfer via philopodia. So what you can see in this image is an example of EC90 beta cell that is infected, and we can recognize this because it is GFP positive. And this cell is extruding this philopodia. This is just a schematic representation of what you can see here. And here we have a beta cell that is not infected yet, but is getting in contact with the philopodia of this other cell. In the second image, you see that this cell that before was not green is actually green, meaning that it is infected now. And this cell is making philopodia itself and moving to reach other cells to infect them. And the first one is red because it's dead. And in the end, also, the second one is becoming red, meaning that it's dying, also this one. And here you have the nice movie of what I just presented. So these cells making philopodia, contacted this one, this is becoming green, moving, and then you would see, okay, now it's rounded, detaching from the bottom of the well, and red, so it's dead. We also measured this way of forming philopodia. We used the measurement of the eccentricity, meaning how rounded is the cell, and you can see that the cells that are infected in green here have a variable shape at the various time points when compared to cells that were not infected, in which the rounded shape is quite stable, meaning that it's not rounded because the ECN anti-beta cells are not rounded, but it's stable over time. And importantly, in collaboration with Sarah Richardson from the University of Exeter, we could make similar observations, meaning that, of course, it's not exactly the same because here we are in vitro, but she could observe cells infected, meaning VP1 positive, in the islet. In this case, it's a tissue from an NPOD type 1D organ donor. And the measurement of the diameter, the area, and the perimeter of these cells confirmed that all these characteristics were all higher in infected versus non-infected beta cells, somehow mimicking in reality what we observed in vitro here. So after observing all these passages that you now know, at the end, what we observed was that all the cells were dying. This is what I explained before. And in particular, we decided to focus on this last step, and we observed that these cells were dying in a single cell pattern. So to explore the way these cells were dying, we built a mask, and for this experiment we used a CVB that was non-GFP because we wanted to do a background staining of the beta cells that was done in red. Then the cells were infected, so they were making philopodia, even if they were not GFP positive, because in this case the virus was not fluorescent. And in the end, these cells were becoming positive for the dead cytotoxic green marker, meaning that we could observe that they were yellow because they were red at the beginning. So we built this mask that was able to identify the cells that were dying as in this single cell pattern. And here you have a movie of an example of a condition in which we put beta cells and the virus, and we acquired images for more than 24 hours for sure, and we applied the mask to detect the cells that were dying, and you can see the single cell pattern. So we were ready at this point to compare the CVB mediated versus the CVB reactive T-cell mediated beta cell killing. So on the one side, we had just what I presented, so the cells, beta cells with the virus, and on the other side, we had the beta cells with the CVB reactive T-cell, CD8 T-cells. So we pulsed these cells with the CVB peptide, and we observed that on the one side, the cells were becoming green, which means yellow, and the mask for the single cell detection was applied. And this is how it appeared in reality. And we observed that on the other side, we had a completely different situation, meaning that the cells, when they were dying because of the T cells, they were dying in clusters. And this is how it appeared in reality. So we were ready to study a complex situation, which means a mixture of the virus and the CVB reactive T cells. So we put some cells, beta cells, we infected them with the virus, and then we put on top T cells. And because of the masks that were developed, we were able to measure how many of these cells were dying because of the virus and how many were dying because of the T cells. And here you have an example of how the two masks were applied in a mixed condition with the virus and a T to beta ratio of 1:1. This is the same field that was acquired. Here you see the identification of the clusters, and here you see the identification of the cells that were dying alone. So this is the representation of what we observed at various time points in terms of increasing amount of CVB killed beta cells on the top part of the graph, and increasing amount of T-cell killed beta cells in the bottom part of the graph. Overall, for all the conditions tested, meaning that the various shades in this case represented the various amounts of T cells, the various ratio, and each graph is representing a different amount of virus, so 10, 100, and 300. So overall, what we could observe was that the CVB-mediated beta cell killing is faster and is stronger as compared to CVB reactive T cell mediated beta cell killing. We wondered whether this was due to this fact that most of the cells were dying because of the virus, was due to the high amount of virus that we were incubating beta cells with. But since the observations of the association between the Coxsackievirus infections and the development of the autoantibodies is based on a low-grade persistent CVB infection, we wanted to test what was the outcome of incubating beta cells with a low amount of virus. So we, as you can see, we used a very low amount of virus on top of beta cells, and we concluded that we just needed to wait more, but that in the end, at 72 hours, as compared to our controls, the non-infected cells, all the cells were dying. And we also confirmed the downregulation of the HLA class 1 on the cells, and as you can see, it's adherent cells, just because we wanted to keep those cells that were still adherent to the flask, because as you saw before, the cells that are dying are detaching from the bottom of the well. So when we evaluated this HLA class 1 downregulation, we could find this downregulation for almost all the conditions that we tested, even if the MOI, the amount of virus, was very low. At this point, we wondered whether the infection of beta cells by Coxsackievirus was somehow affecting the killing mediated this time by autoreactive CD8 T-cells, meaning that so far what I have presented is based on the use of CVB reactive CD8 T-cells. But in this case, we wanted to test what was the effect of putting on top of beta cells PPI TCR transduced CD8 T-cells. And of course, we could observe that in absence of the infection, these PPI TCR transduced CD8 T-cells were already killing the cells. But because PPI is already present on the surface of beta cells. But we observed that even at a low MOI of 1, we could see an increase in the killing of beta cells mediated by autoreactive CD8 cells upon CVB infection. So on the one side, we studied the effect of Coxsackievirus infection on beta cells, and on the other, which were the characteristics of the T-cell responses mounted after CVB infection. And to summarize what you can see in the paper, we observed that only few CVB peptides are presented by infected beta cells, which could be linked to the fact that we observed a downregulation of surface HLA class 1, that the anti-CVB CD8 T-cell responses is restricted to even fewer of these peptides, that the cross-recognition between a CVB peptide and the GAD epitope is at play for CD8 T cells, that the CVB reactive CD8 T cells are near absent in islet infiltrates, that the Coxsackievirus transfers through philopodia in beta cells in vitro, and that still in vitro, the beta cell killing is mainly CVB mediated. And the last data is about the cytotoxicity of PPI reactive CD8 T cells against the CVB infected beta cells, which is enhanced upon infection. So if we want to present some possible implications from vaccination, starting from what we observed, our data suggest that among the three scenarios that I presented at the beginning, the first one is the most likely to happen, characterized by a poor CVB antigen presentation and a poor anti-Coxsackie-virus immune response, which suggests that CVB vaccination may be effective in strengthening this weak immune response and limiting viral propagation. Regarding the epitope mimicry mechanism, even if I presented one of the cross-recognition that is actually at play, the peptide the peptide presented is not included in the vaccine. So it might be at play for the infection, but not in case of vaccination. So, which are the perspectives? Coxsackie-virus infection and type 1 diabetes. So far, I have presented to you what is included in the paper, and now I'm going just to give you an introduction of what we are doing now, and we are going to do. So, we wanted to characterize the differences between the immune response in a natural CVB infection versus vaccination, meaning that in case of the vaccination, we have only P1. In case of the infection, we have P1, P2, P3, meaning that we can distinguish the response between those that are getting natural CVB infection versus those that are getting the vaccine. And we are currently working on this, exploring not only CD8, but also CD4 T-cells. And regarding the study of the immune response against the natural CVB infection, we are working on the deep longitudinal samples from which we have data about data from the sera, from the stools, and on which we will study the T-cell, we will perform the T-cell assays to study the correspondent response in PBMCs. And this will be done at various time points. And then we are about to investigate the effect of CVB infection and the CVB reactive CD8 T-cell killing on pancreas slices, because as you saw, what we built is methodology that is quite simple, that is not reproducing all the complex components that are indeed in the human body. So by working on pancreas slices, we wanted to go closer to what is happening in reality and to understand whether what we observed in our simple model is also reproduced on a much more complex environment. And we are about to investigate further the CVB-GAD cross-reactivity, because we observed that this cross-recognition is at play for CD8 T cells, but we are trying to sort some of these cells to sequence the TCR, to re-express this TCR, to understand whether T cells expressing the TCRs can be cytotoxic against infected but also non-infected beta cells. And then the last 2 points are the focus on the interplay between the innate immune cells and Coxsackie-virus, and this will be focused on the pancreas, so in particular the role of neutrophils in the pancreas upon infection, but also at the enterocyte, so meaning the role of neutrophils on CVB infected enterocytes. And on this side of the enterocytes, that is there is also an entire project that is ongoing, and this is the PhD project that is currently ongoing, performed by Orlando Burgos Morales, PhD candidate in the lab. And we already have some data on from CVB infected enterocytes. So for instance, we know that we observed the same HLA class 1 downregulation, but we observed instead an HLA class 2 upregulation upon infection on these cells, which might suggest again the importance of involvement of CD4 T cells in this picture. Also that they are more resistant to die, and they don't die, so they don't die easily when put together with CD8 T cells recognizing the CVB peptides, and that also in enterocytes the CVB transfers through philopodia. So this is what we are going to develop, and any question or comment or a suggestion is welcome. I'm going to thank all our collaborators and all the people in the lab, and thank you all for your attention.
Thank you very much, Federica. The beautiful and elegant work and study. And you've got a couple accolades in the chat as well. Just really so exciting that you really used, as you said in your own words, a simple system to kind of dissect this big question, and informed, because I mean, it really will inform vaccine development. And I loved this whole piece about the Philopodia. I think that's so interesting. And people like Per Ola Bergen and Karolinska, he's kind of documented the whole Philopodia that the delta cells put out just sort of in vivo. And it will be so interesting that you guys are now going to go into slices and look carefully at what's happening there. And I wondered if anyone has thought about or looked at the CVB infection or interaction with the other cell types, in that context. If you're talking about sort of the beta cells putting out these Philopodia to infect other cells, I mean, I wonder if they're going to be, if you'll see any kind of infection of its other neighbors, the other constituents of the islet. Do you have any thoughts about it?
I think it's very interesting to explore this world, and actually it's not only related to the components of the islet or of the pancreas, but also I'm planning to understand to understand the role of other components of the immune systems, as I anticipated in the last slide. I'm going to explore more for instance, the role of neutrophils in this, and we have some preliminary data suggesting that actually neutrophils are able to uptake the viral particles, but this cannot, this is apparently not able to actively produce viral particles inside the neutrophils, so no replication at place, but uptake, which might mean that this might also might drive the movement of these viral particles. So I'm at the moment, what I would like to do would be to explore further, not only, as you were saying, not only the idea of the Philopodia in the beta cells, but more broadly on what is going on when we have a cell that is exposed to CVB. So as in the as for the enterocytes, they are also making Philopodia. This is what we observed. So it seems to be something that is not restricted at all to beta cells, and I'm not surprised. I think it's a way the virus try to escape, to stay hidden inside the cells. So we might think also about the patchy appearance of the VP1 positivity. This might be related to the fact that the cells really need to stay in contact. And actually, among the various experiments in our simple model, we also put non-transduced primary CD8 T-cells, and only because they were in between, the cells were getting infected less. So I think that this mechanism of forming the Philopodia is very important. We need to explore which cells are at play in this passing the virus.
Yeah, and it's interesting. And, I mean, in vivo, they've got the beta cells are connected to other cells in the islet via their gap junction. So that's another access route. And you might think, okay, well, who knows? I mean, once a virus is on board, maybe polarity is lost and junctional complexes are screwed up, and then a Philopodia develops. So it'll be so exciting. And you guys have all the really great technology already in place. Sarah Richardson's group and Teresa Rodriguez Calvo's group, and it's really going to be sort of exciting times to see what comes out of the next steps. I wanted to open it up to anyone else. Oh, I think I see. Sorry, this is a little bit. Question from Yang Dai. Do you want to unmute, Yang? No? Okay, I'll ask it. What causes the downregulation of HLA-1? Because the cells are dying or some molecules of CBB?
But we are not the first observing this kind of mechanism upon viral infection. The fact that you have an HLA class 1 downregulation. So first we have an impact on the trafficking. So the anterograde is not anymore there trafficking, but it's going to be a retrograde trafficking. So the fact that we have less HLA class 1 molecules on the surface and the less peptides presented, this is kind of expected. It's not related to the fact that they are dying on enterocytes. They are downregulating the HLA class 1 and they're not dying there. So it's a direct effect of the viral infection. Then we need to consider that in reality, in the human body, this might happen in a completely different way, because once we have the production of the interferons, and actually we tested this in our system, and there is no interferon alpha or beta production by the beta cells upon infection. So when we have the interferons at place, this MHC class 1, HLA class 1 downregulation can be reverted into an upregulation, and then the peptides can be presented. So probably this is not really representing what is going on in the human body, because it's a much more complex system. But this is how the virus is working on the cells. So this is, I think, in any case, an important point.
Yeah,
I'm wondering, anyone try to targeting downregulating class 1 MHC by the CPV to prevent the disease?
To target, sorry?
Because your CPV can downregulate HLA class 1. Does anyone trying to use this pathway to treat the disease or some prevent the disease?
The disease is characterized by the presence of interferon-stimulated genes. I would not expect any clear HLA class 1 downregulation in the diet. So we all know that there is this interferon signature and the pre-type 1D already patients. So I think that since we need to consider that this might be at the very beginning, so months or years before the clinical symptoms, I think that this is too early to intervene directly on that. I think it might be of interest, but I think it's not feasible because of the timing. We still don't know much about the timing of when, how, and when this is developing. So first, we need to focus more on that. And once we dissect the exact timing, which might be different from one person to another, I do expect that, then we can try to target the downregulation. That might be great.
Thanks for that question. Any other questions? Please feel free to unmute yourself or drop something in the chat. In terms of the neutrophils,
Yes.
Are there already sort of repositories where you can pull from to kind of get some data around that whole neutrophil landscape in these patients in the different stages?
But in the different stages of type 1D?
Yeah.
Yes, this is part of my previous work in the lab of Milan, and in a collaboration that we performed with a group in Canberra. And basically what we observed was that in any case, neutrophils are abnormal, and they are abnormal already before the onset of clinical symptoms. And by isolating them, we could observe that there was this signature, so interferon stimulated genes that were upregulated in already in a pre-type 1D first degree relatives. This was part of the samples from TrialNet at that time. And together with this, we also obtained the information of observing neutrophils that were aggregated with platelets. And this is another paper that came out on JCI Insight, and this was done in collaboration with Canberra. And the fact that they were together with they were found more aggregated with platelet might predispose them to have a priming and to extrude net, or to be in any case activated and ready to go. Then I have to say that I think that there is a huge work to explore now on the CVB neutrophil side, because there have been a lot of publications on the side of myocarditis. So I think that now we have a lot of information on neutrophils, how they can act on, for instance, on the CAR receptor in the intestine. This has been published already, and directly on the on CVB, meaning that as I was saying, they can uptake, but the virus cannot replicate, but they can also play the role of Trojan horses of the virus.
Yeah. How feasible do you think it would be to use neutrophils as a biomarker in the clinic?
As they were found to be abnormal already at the beginning, I think that it can be harder to use neutrophil alone as a biomarker. I think that it's something that has to go together with something else. So I imagine more something like a complex of biomarkers that can be used. So something that could target, for instance, neutrophil elastase, or but this together with the prevention of the infection. This cannot go alone. That's
right. It would have to be kind of a more complex testing on the back end.
Yes, I guess so.
Yeah. Okay, so just a little pitch for the T1D TCR genetic data repo that we're building. It's at 80 million sequences now and should be at 1 billion by third quarter this year. So any of these CD8 positive T cells that are recognizing the structural and non-structural CVB epitopes, that they provide biomarkers to differentially follow response to vaccination and infection. It would be really great to have some of these data now, or as it progresses, deposited into this growing T1D TCR data repo. And our monthly working meeting occurs later today at 1 p.m. Pacific time. Should anyone like to join, just drop your email into the chat to learn more. Thank you again. Just fantastic work, and we're really excited to see what you and your team does next. So thanks again for joining us, Federica.
Well, thank you for this opportunity, Monica. It's been great.
Okay, talk to you later. Bye-bye.
Bye-bye.
Bye.